And now we come to the most difficult part here, the human being, as always. And we will have a look at the observer. First, it's interesting to know that the spectral sensitivity of the eye is changing a lot. So from 400 to 700 nanometer, that's the visible range of the light here from about. And actually, the brightness function is different. So yeah, it's different for different wavelengths here. It's greatest in the middle, and that's for the greenish parts. So as we come from the woods, I think that's why we are can differentiate mostly in the greenish part. And for example, you can see that also if you have a red light with the same energy as a green light, that red light seems much dimmer to us. So objectively it isn't. So now first, what is happening in the human eye? So the light-sensitive sensory cells of our eye are located in the retina here, and onto which the light falls. So the light falls through the lens and falls onto the retina. And the sensory cells are divided into rods and cones. So the rods are only for the perception of light and dark. And concerning the cones, we have 3 different types. So one are for perceiving blue light, the others for red light, and the third one's for green light. So we have only these 3 sensory cells. And for example, you see there is no yellow cone, no sensory cell for yellow. That's actually what's happening in the human brain then, in the combination of the impulses of the 3 cones here. And now to translate that into something objective, there was a big experiment done with many people here to quantify this sensitivity of the human eye. So this test was carried out with many different test subjects, many different people, and corresponding to the cones in the eye, red, green, and blue light sources were used for it. So how was the setup now? We have a screen here, an upper part and the lower part of a screen or backdrop here, and it's parted by a black partition. And first we have a test light of a certain wavelength, which falls onto the lower part of the screen. And then the person was set behind a reduction screen so that the angle at which he looked at the screen was exactly 2 degrees. I will explain later on why 2 degrees. So, but the same situation for all the people then. And this person was asked to create the same color, the same yellow as at the bottom here on the top part of the screen. So they could control these 3 lights, so blue, red, and green, and then change it until they say, okay, it's the same light in the upper part as in the lower part. And this then was recorded as a function. So for example, if you have here 460 nanometers, that is the light which was sent on the lower part of the screen, and the person selected to have a very big portion of the blue light, a little bit of the red light, and very low portion of the green lamp here. So these are these 3 points which were received at 460 nanometers, and that is done for every wavelength then. And then you get curves. So 1 curve for the blue light, 1 curve for the red light, and for the green light. And these 3 curves or functions are called the 2-degree Standard Observer, and this was done in 1931 by the Commission Internationale d'Éclairage. Okay, so in this way we had an objectivity for the observer now. And then I said I would tell you about the 2 degrees. One selected the 2-degree opening angle as one thought this fovea here, the viewing pit, is the area where the most sensory cells are found. So that was why they selected the 2-degree opening angle. But later, actually, it turned out that the cones are also densely distributed beyond the fovea. So this experiment was repeated, and then with a 10 degrees opening angle. And now the area here is the whole yellow spot, or macula lutea, it's called. And that's, yeah, normally the approach we're using. So if you're doing color measurement, you can select between the 2-degree and 10-degree.
Yeah.
Do you have a question? Okay, just a moment. I think I have to mute that. No, it's okay. Okay, you can select that, for example, for traffic signs, one uses still the 2-degree opening as they are far away, and thus the 2-degree is really reflecting that position. But normally one is using the 10-degree angle nowadays. So that was done in 1964, the repetition of this experiment. And you can see, so that's now the 10-degree standard observer. And so the whole setup was a little, if you see, it's different. If I have a 2-degree opening angle, I have only this small area that I can see. And if I use a 10-degree opening angle, it's a much wider screen I can see here. And of course, now we have 2 standard observer functions, or actually 2 times 3 standard observer functions. And this is the 2-degree standard observer as we saw it beforehand. And now we have a new one, the 10-degree standard observer, which is slightly different, as you can see here. Especially here, it differs a lot. Okay, are there any questions here? Please be brave, because it's not easy, I would say. Okay. Yeah, well, then actually we have solved the third element here. So the observer now is quantified by the standard observer function. So this way we now really put the 3 subjective elements of human perception into 3 objective elements here. Okay. So we are done for the observer as well.